In this video, let us discuss about Meristem culture. So, before entering into the Meristem culture, firstly you have to know what is meant by Meristem. Meristem. So here meristems will be of two types: apical meristem and lateral meristem. Okay. So here the apical meristem is a tissue in most plants containing undifferentiated cells found in the zones of the plant where growth takes place. So undifferentiated cells are nothing but the meristematic cells only. So by this definition you can understand that where does this meristem present? Meristem will be present at the region where the growth occurs. For example, if you see, even this apical meristem will be also of two types because they are present at the root region and they are also present at the shoot region. Root and shoot. Root system and shoot system is nothing but the root system is nothing but which is present below the soil, and the shoot system are the parts which are present above the soil. So these roots are present, as we know that the roots are present in the soil. They are called as root system. As these shoots, that's nothing but the stem, branches, leaves, etc. Flowers, fruits. All of this will come under the shoot system only, which are present above the soil. So if you see here, in this case, the root apical meristem and shoot apical meristems are the two types of this meristems, apical meristems. Okay, so what I have said to you, these apical meristems will be present at the regions where the growth takes place. So if you see here, at these apex regions of these roots, their meristem will be present. So why it is present? The only why in this only region this meristem will be present, because at this region only the growth of the root occurs, right? So at this apex region, this sharp end region are known as apex, hence they are called as apical meristems. Okay, so this apical meristems will be present at the edge region of the root. Why it is present at the edge region? Because the growth normally occurs at that position only, right? So these meristems will be present at this edge region only where the growth occurs. So here also, if you see in the case of shoot system, and the shoot apical meristems will be present at the edge of the shoot. Why it will be present there? Because the growth occurs from, the growth begins from there itself only, right? So that's only the region. The meristems are present at the region where the growth occurs, where the growth begins. Okay, so that is only the definition of this apical meristem, and this will be your lateral meristem. Okay, so the this whole is said to be as lateral meristem because at this region the total growth also occurs. So the name itself, it indicates that the lateral meristem is nothing but lateral. Okay, so normally basically this is about the meristem. So let us discuss about the meristem culture. So what is meant by this meristem culture? Okay, so I have said you that this is a meristem, right? So normally that meristem will be extracted. I mean, for example, if you take shoot apical meristem, this will be extracted. And even in this case also, I'm taking only the shoot apical meristem only for your better understanding purpose, and this shoot apical meristem will get extracted, and this shoot apical meristem will undergo culturing process. So you know about the plant tissue culture, right? So if you know about the plant tissue culture, then only you can understand this meristem culture, and you can know the technique also. So I know even in this video also I'm going to explain you the technique, but it's better to know about the plant tissue culture also. Okay, so this shoot apical meristem will undergo that culturing process, and when it undergoes a culturing process, the finally a product will form, and that product will be also a plantlet, and that plantlet which has been formed finally will be with free of virus. So how the plant will be produced? So that's the phenomena of this meristem culture. So if you see the definition of the meristem culture, the technique in which the production of virus-free plants will be obtained from the mother plant by taking its shoot apical tip as an explant. So you know about the explant. What is meant by explant? Basically, it is a part of a mother plant. It is a part of a mother. You see in this case, this will be your explant. Why it is called as explant? Because it is one of the part of your mother plant, because this bee will get extracted. When this will get extracted, that is called as mother explant. So this will be your explant because the shoot apical meristem will get cutted off. When this shoot apical meristem will get cut off, then immediately it undergoes culturing process, which we will do. Okay, so this will be your explant. As I have written here, the shoot apical tip as an explant, the technique in which the production of virus-free plants will be obtained from the mother plant by taking its shoot apical tip as an explant. So by this definition, it is clearly understood that here the shoot apical meristem will be isolated, will get extracted from this total mother plant, and that shoot apical meristem will undergo culturing process and forms a virus free plant. Okay, so how this process will be occur will be explained you later in the diagrammatic explanation. Okay, so coming to the discovery, there are two scientists called Morel and Martin. They discovered the total technique of this meristem culture in 1952. And later, Murashige and Skoog in 1962 explained that there are totally three stages which will be involved in this meristem culture. So let us learn what are those three stages. So coming to the stage 1, explant may develop into single shoot or multiple shoot, and in this stage, what happens is that it requires some of the chemicals for its energy purpose, and those chemicals which has been required are cytokinins and kinetin. This will be a growth regulating hormone, and this kinetin is a protein which will be utilized in the stage 1 process. And coming to the stage 2, multiplication of propagule. Okay, and with this, in this multiplication of propagule, for this axillary shoot proliferation is followed. Why it is followed? Because it maintains higher genetic stability. So you remember students, higher genetic stability is very much important when you perform a plant tissue culture. So this axillary shoot proliferation takes place. So this will be your stage 2, and in this stage 2, high levels of cytokinins are required. This hormone, the cytokinin hormone, is very much required. Okay, so you know about the plant growth hormones, right? There will be three types of plant growth hormones: auxins, cytokinins, and gibberellins. Among these, cytokinins will be required in a high levels. Require be in high levels. Remember, high levels in the stage 2. So now coming to the stage 3, regeneration of adventitious roots from the shoots. Okay, roots from the shoots. So by this you can understand that so if, for example, if you don't think this is a mother plant, just think that this is a thing which you are going to produce. So here firstly the shoot will be produced. Okay, remember one thing, when you do meristematic culture, only first the shoot will be produced and later the root will be produced from the shoot. So you can understand this sentence when I'm going to explain you the diagrammatic representation. So please wait for it. Okay, then you can understand this stage 3. So by these 3 stages you can understand that the total plant will get developed at finally as a product, and the product which has been formed is a plant which is free from virus. So how it will become free from virus will be given in the will be explained properly in the diagrammatic representation only. And remember, in this stage 3, what are the energy source which are mainly which plays a major role in the stage 3 are NAA. That's nothing but naphthalene acetic acid, indole butyric acid, which is shortly abbreviated as IAA, indole acetic acid. And here one of the hormone which is mainly used in the stage 3 are auxins. So in this both cytokinins will be used and here auxins will be used. Okay, so now let us see what are the components which are mainly used for this meristematic culture. Okay, inorganic nutrients are used, organic nutrients are used, and make sure that these organic nutrients should be rich in vitamins. Okay, amino acids, plant growth hormones, and solid fatty aging cell. All of these are the main important things, components which are mainly used in this procedure or a protocol of this plant tissue culture. So now let us see the diagrammatic representation, and for your practical purpose